very much, and thanks for the opportunity to talk to this group. What we'll be doing is talking to you about a lesion which I think sort of understanding of this lesion sort of changed the course of the way we look at transplants. Just set my stopwatch here. So this is still an important lesion. These are my disclosures. So these studies are supported by a lot of different grants, and we still manage to keep it going. So I'll be talking about what is tubulitis and the history of tubulitis and its key role in the Banff classification that the world uses, and then the molecular associations of tubulitis and of T-cell mediated rejection, and then how time changes T-cell mediated rejection, and above all, how this impacts nephron injury and how the nephron feels about all this. So I'll give this introduction, then Kim will talk about Banff and the fundamental discoveries in tubulitis. I'll give you the current picture, and then Majid will come in showing you how we're using logistic regression in his graduate student project. Okay, so this is an important lesion. In the 1970s, Kim will tell you how it was first described in renal disease and then redescribed as occurring in what was then called rejection. Antibody-mediated rejection wasn't recognized. At that time, I was seeing cases in Toronto and then in Edmonton that seemed to be different and seemed to be associated with antibody and possibly antibody-mediated rejection, but nobody would accept that this existed. Kim and I arrived at the U of A in 1987, and we started working together, or in parallel, more or less. It became clear that the cases of possible antibody-mediated rejection had accumulated in Toronto had no tubulitis. So in 1990, we were able to publish the first paper, which said that there were really two rejection phenotypes. There was a microcirculation process, which has subsequently become the dominant rejection phenotype in all of transplantation and all organs. And then there was a tubular interstitial process, T-cell mediated rejection. And this set the stage for Banff 1991, and set the stage for our molecular project, because we were then able to model the T-cell mediated rejection as a specific process in mouse that also had tubulitis. So we modeled it in knockout mouse. We used microarray results from the mouse to help develop the human study. And so then we developed this genome-wide assessment of human biopsies. This is the paper. And the paper really, the key point in the paper is that the cases with antibody-mediated rejection had microcirculation changes, but did not have tubulitis. Both types could have V lesions, and the ambiguity of V lesions has been a puzzle since that time. So that was 32 years ago. And subsequently, Kim Solez has gone on to become really the world leader in transplant pathology of all kinds. And fundamentally, it's the organizer of the way that consensus is reached about these processes and diagnosis. And the Banff system, then because we had the MAP system, we were able to take that to the FDA when we were starting immunosuppressive drugs. And they agreed that we could use a biopsy-proven rejection as an endpoint for the immunosuppressive drugs. And all the immunosuppressive drugs you're using were used with that endpoint, because we could convince the FDA that we had a reliable way of making diagnoses. So Kim, over to you to tell us about tubulitis.
Okay, so if you want to unshare, and I will share, yeah, there we go. I'm very pleased to have this opportunity. It's less than a month since I spoke here before. I am very grateful, and it's very nice to do this joint presentation. So Banff, fundamental discoveries and tubulitis. On December 6th, 2021, something very surprising happened to me. Phil contacted me with a simple question: who identified tubulitis as a marker of TCMR? And he said he learned about it from me, and he believed it to be a fundamental discovery, and it even outperforms many of the gene transcript sets as a predictor. So I was pretty excited about that. I happen to have this big stack of editions of Heppenstall's textbook here, and so that's where I started, and it was very clear that edition after edition had nothing about tubulitis in the early days. Ken Porter, who wrote the transplant chapter, simply mentioned finding lymphocytes in the urine, but tubulitis was not described. So when you think about fundamental discoveries, what do you think about? I think of the Higgs boson, but that's kind of old, you know, it's 10, 11 years old. And it came about from the Large Hadron Collider, which is what's shown there at the top. This is something I'm currently involved in, but it will impact all of you, probably surprise the heck out of you. There is no standard method for indicating whether you're dealing with a human, an AI, a human guided by an AI, or an AI guided by a human. So 20 of us are creating the standard for this. And I think I'll end up either the vice chair or the secretary of that group. And it is, I think, a kind of fundamental thing. But let's get back then to the fundamental discovery of tubulitis. As I've said, the first 4 editions of Heppenstall's book contain no reference to it, just talking about seeing lymphocytes in the urine. And they said that in more active rejection, there were bigger lymphocytes. So that's what we tried to depict here. But otherwise, it is remarkable. I don't know how they think the lymphocytes got to the urine, but they did not really look at the tubules. So tubulitis was first described in 1975 in the native kidney and given that name. And the definition at that time included not only invasion of the tubules, but also inflammation around the tubules. And Vic Pollack had been talking about this in teaching since about 1972. Richard Sibley, trained at the same institution, heard Vic Pollack talking about it, and he was the first person who described tubulitis in the transplanted kidney, 1983. But his definition is very precise, I would say, and wrong. It makes it sound like the lymphocytes are inside tubular cytoplasm mainly, which actually does not happen. It occasionally happens, but it's pretty rare. So that was in 83. In 84, Regina Varani described it with this vague phrase: mononuclear cell infiltrate into the tubular epithelium. And in 1985, I described it as mononuclear cells lying between tubular epithelial cells or beneath them, just inside the tubular basement membrane. Now, when we wrote the first Banff paper, published in 1993, it was very interesting that the configuration of the paper was heavily influenced by the editor of Kidney International, Tom Andrioli, who believed that tubulitis was such an important concept, and the non-specificity of the interstitial infiltrate was also so important that everything about that should be on the first page of the article. Because he argued many articles, people don't get beyond the first page. So you want to put the important stuff there. And that was done. And it has served us very well. So a lot of people who might have passed by this got to read this particular paragraph, which you can see begins with tubulitis and ends with tubulitis. Yeah. And today and into the future, tubulitis remains just as important as ever. It figured prominently in the 3 articles in the March 2022 issue of AJT. The cover graphic you see here, acute T-cell mediated rejection, still a worthy opponent. And for decades, tubulitis has been the focus of morphometry programs. So now the best machine detection systems use a combination of handcrafted features, texture analysis, and machine learning. And we have a really intriguing recent video from the Banff Digital Pathology Working Group with Ulysses Bayless showing this very, very well. And you can imagine that in the future, there will be a lot of automatic detection of tubulitis. This will be very much a thing. Now, in Phil's talk, he's going to tell you that to a certain extent, tubulitis continues even after renal function comes back in a rejection episode. And it may surprise you that a lesion would stick around after function has returned, but actually that's very common, particularly in the area of acute tubular injury. This was the best known article that I had written at the time that I came here. And what it shows is in acute tubular injury, there are 12 lesions, and 10 of those 12 persist after functional recovery. Only 2 of the lesions correlate with function, were present when renal failure was present, and absent after recovery. And they were thinning of PAS positive brush border and shedding of individual tubular epithelial cells, leaving bare basement membrane. So it seems like functionally, those are the only 2 important lesions, but the others are much more picturesque. So even now, we have biopsies from patients who happen to be biopsied after function has come back, where there are spectacular lesions of which you would regard as acute tubular injury, but that would not include thinning of brush border and shedding of individual tubular cells. So that means if you lump all the lesions together, you don't get anything useful, because 2 of those lesions are highly meaningful in terms of the cause of renal functional impairment, and the other 10 are not at all meaningful in causing renal functional impairment. Anyway, Heptesol called that paper the best that can be done with a difficult subject. And later, I looked at the same questions in the transplanted kidney, an important antecedent to Banff's studies. One of the interesting things we found there is there was more true tubular necrosis, which you seldom found in the native kidney, but relatively little brush border loss. So the morphology was somewhat different. Yeah. So anyway, that's what I wanted to tell you. And now I think Phil can go back to his presentation.
Yeah, I just wanted to say how grateful I am to Kim for all he's done in the field as a clinician. I think he set the stage for us to develop immunosuppressive drugs, and the immunosuppressive drugs really changed the outcome for many patients.